how to work. But I have a grader, graduate student helping me, so it's great to have a teaching assistant for this course. Okay, so drilling down a little bit into molecular biology is what we're going to be doing in the next few chapters. And this chapter is mostly about DNA replication. And really want to treat this in a manner different than other courses you may have had on the topic, where we're just going to be focusing on the little biomotors and bioengineering in terms of the enzymes that carry out this incredibly complex process. So the names and basic functions of the catalyst is my focus. And then we're going to be doing some comparison between eukaryotic and prokaryotic systems. You've studied eukaryotic systems, and we'll contrast the prokaryotes. So terms, understand that the genome represents all of the DNA that's inside a cell. And so that's going to include extra chromosomal components like the plasmids. And plasmids are small molecular weight circular DNAs. Some of them are pretty large molecular weight. You can get some mega plasmids that are 200,000 base pairs. That's a big plasmid. But nothing like 2 million base pairs in the chromosome. And so like I said in the early part of the course, typically a bacterial cell has one chromosome. Some of them have two. And the appreciation here is, as I've said before, just even this one chromosome has got to be kept really tightly packed and organized inside the cell, because there is one chromosome that not packed tightly. And if you would line that chromosome end to end, what does it say? 1,700 times, 1,500 times the length of that cell. So a massive amount of DNA is packed inside that cell. Okay, so we all are used to vertical genetic transmission. So mother and father vertically transmit our DNA to our offspring into the next generation. So that's vertical transmission of DNA, of genetic information. But bacteria have horizontal genetic exchange as well as vertical. And so the horizontal is transfer of DNA from one cell to another in the same generation. And then some terminology in how we organize genetic information, or organize discussion of the genetic information. The structural gene produces a functional RNA, and that usually encodes a protein. But we are in the midst of a pretty good set of discovery processes where what had previously been thought to be dysfunctional DNA with no function we are uncovering more and more function to that DNA. And so when we say usually encodes a protein, there's a lot of genes that don't encode protein but are functional nevertheless. So we'll talk a little bit more about that. But as far as a structural gene that codes for a protein, that's the traditional definition of structural gene. There typically are upstream of those structural genes promoter sequences that control the expression of that structural gene. So DNA control sequences, promoter regions, are these regulatory DNA sequences, that regulate the expression. And these are never translated into RNA. They're just controlling whether that structural gene is expressed, is transcribed into RNA. Okay. This is kind of an appreciation table. There's no real specific information that I'm after here, but it's a pretty good overview of giving you example bacteria. And first of all, the size of their chromosome. So most of them are circular, and we are looking at this in terms of kilobase pairs. So when you see 4,400, that is 4,400 kilobases. So you add 3 zeros to these numbers, and you get the genome size. So a genome size with 4,400 kilobase is 4.4 million base pairs. And that's typical of bacteria. You can say bacteria have between 1 and 5 million base pairs. They're smaller, and there's some larger, but between 1 and 5 is a good kind of average. So here is Burkholderia has 3 chromosomes. And I would probably count this as a large plasmid. But 3 circular chromosomes. Here's a large circular chromosome. Here is the organism that causes Lyme disease. Borrelia burgdorferi has 0.9 million base pairs, and it's linear. And then Agrobacterium has a chromosome and then a linear chromosome. And those definitely are 2 chromosome base sizes. That's not a plasmid, that being more than 2 million base pairs. Okay, and then just some representative archaea, just for your information. And then understand, we're going to talk about plasmids a little bit. Some bacteria have plasmids, other bacteria don't. So they typically carry genes that you absolutely don't have to have, but they sure come in handy when you're exposed to antibiotics, or they sure come in handy when you're exposed to heavy metals. So they give you traits, they give the bacteria traits that are really helpful under some conditions. Okay, so that's overview, figure, no real specific information there needed. Okay, so there's your genome size range, ranging from 500,000 to more than 9 million base pairs. And then compared to eukaryotes, we are typically in the billions. So 3 orders of magnitude greater base pairs do we have than bacteria. So they're in the millions, we're in the billions. That's about the resolution that I'm kind of stressing as far as these numbers. So 3 million kilobases is a total of 3 billion bases. Bacteria have 3 million bases. So 1,000 times more. But you start thinking about complexity. Well, we have 1,000 times more because we're, actually billions of times more developed than a bacterial cell in some ways. But genome size doesn't directly correlate to, well, like intelligence. The potato has more nucleic acids, more base pairs than the human being. So, the IQ of a potato, Mr. Potato Head's pretty smart, but not where Homo sapiens is. So that's kind of one interesting thing. And we'll talk a little bit more about, well, what is correlated with sophistication? Well, it's, quote, junk DNA, which is what people used to call the presence of non-coding DNA. So this is DNA that doesn't translate into a protein. So a lot of it stops at RNA. And in the past, people have thought that's just a bunch of junk, and it's been totally not true. A lot of that DNA is very functional, and so I don't think junk DNA is used anymore in your textbook. Okay, so here is an interesting thing, is eukaryotic organisms, actually greater than 90% of our DNA is non-coding, does not code for a protein. What is the function of 90% of our chromosome? that's a question that is being answered and will be answered, in 10 and 20 years. It's really a very, very interesting and very important DNA. 10 years ago, people had no idea, and they just called it junk, which was dumb. Just because you don't know what the function is doesn't mean it's without function. Okay, so if anything, the sophisticated eukaryotes have a lot more non-coding DNA, greater than 90%, whereas bacteria have less than 15% of their chromosomes being non-coding. And especially when we talk about genomics, we'll talk about more of that. Okay, so that's kind of introductory material. Hopefully whetting your appetite for some interesting things in the chapters ahead. Okay, so genome organization. Here is a real prototype. This is a real model setup in the prokaryote. Here are 3 different genes that are actually transcribed and translated at the same time. And so here are your structural genes. And then, get used to upstream is to the left of your structural genes are these promoter regions, are these regions of DNA that control whether those structural genes are going to be expressed. So that's one level of control shown in A, and these are controlled by regulatory proteins. So here is an even more global level of control, and that is here you have a regulatory protein right here in the middle, a control protein that is actually controlling a number of operons. And when we say operon, this is basically what we're talking about, is a group of genes that are transcribed at the same time. So here's a regulatory protein that is controlling, in this example, 4 different operons. And so when you have the control, the regulatory control of more than one operon, you call it a regulon. So a number of operons are controlled by that same gene. So that's generally true in our chromosomes, though our genes are usually not all lined up as efficiently as bacteria. So bacteria get right on the program. Remember, they are a baby, and then they are making babies. They've gone through awkward teen years, and then they're having babies in 15 minutes, 15 minutes after they were born. So they have to get with the program. And that explains at least some of the efficiency of the bacterial chromosome. Okay. I love the topography of the double helix. So the physical shape is what we're talking about here. And when you have a double helix, it forms this big groove and then a small groove called the major groove in the double helix and the minor groove. And what this is important for is these regulatory proteins. That's their docking point on the DNA. And so those grooves are sterically fit for a regulatory protein to fit into. And so in the second part of this figure shows the CRO protein, which we'll talk in the days ahead about a number of regulatory proteins, not so much the CRO protein. But just to show you that there's this really lock and key type of relationship between DNA binding proteins and the DNA. Okay, so the nucleoid, as I've said, is the region that the DNA is tightly packed within. It's not bound by a membrane like it is in our cells. Bacteria do not have a nucleus. But nevertheless, how does it control that massive molecule in a limited space. It's basically with these anchoring proteins that these are DNA supercoils. So that's a special kind of coil that's very, very tightly coiled. And then the coils are all held together with these, they call them histone-like proteins, because we have histone proteins and bacteria have, well, histone-like proteins that are related in function and even DNA sequence. And they both do the same thing. We have histone proteins that keep our cells, keep our DNA controlled. So that's just a little bit more of information. Here is an unwound, a supercoiled DNA that has been unwound to kind of show you, the effect of supercoiling. And that happens if there's a nick made in one of those strands. Okay, so how does the cell get its DNA into this supercoiled position? So we're going to talk about two types of these enzymes. They're called topoisomerase, and they both change the topology, the physical shape of DNA. And so here's kind of an analogy that's used in your textbook. So the telephone has the old-fashioned telephones with the landlines have the telephone wire, and it is in a coil. And it is, it's easily moved around, but you use it for a year, and it actually kind of forms into what's called, nobody calls these supercoils. The analogy is supercoils because it is now like triply tight. And so does anybody notice that if you unwind your telephone coil, they're all tightly bound. So, the detective Monk walks into a room, looks for supercoils, and goes and relieves them because he's got that fixation. Anybody watch the show Monk? Yeah. So he can't handle those sort of things. And I've got friends of mine that look at my telephone, and until they take care of my telephone, then we can have a good conversation. But until then, they're distracted by things like that. So that's a helpful analogy to help us understand the difference between a coiled telephone line and then a supercoiled telephone line. Okay, in figure 7.9, we have a topoisomerase, and we're going to look at the details of this, that actually breaks the DNA molecule and then moves the other part of the circle through that break and then reanneals. And so in the end, that is the incorporation of a single coil. And that's the function of an isomerase. But let's look at it a little bit more specifically. The difference between type 1 and type 2 isomerases. Basically, type 1 only cleaves one strand of DNA. Type 2 topoisomerases cleave two strands of DNA. So the difference between cleaving one and two strands is the difference between those two enzymes. Typically, type 1 isomerase are relieving tension, undoing coils, and so no energy is required. Whereas type 2 topoisomerases are introducing coils and typically introducing tension into the DNA structure, and so therefore it requires energy. Type 2 isomerases, a good example is the enzyme DNA gyrase, which we will look at in the function of replicating the DNA. So, relieving tension versus introducing tension. And I'm going to kind of make an adjustment on that in about 15 minutes. Okay, so here's type 1 topoisomerase. So here you have a 6-coiled DNA molecule. It does its number, and now you've got 4 coils versus 5 coils. You start off with a 5-coiled DNA molecule and you end up with a 4. So it's relieving the tension, and basically we just saw the mechanism. It cleaves one of the strands and then reanneals, and then allows for the unwinding of one of those coils, giving you 4 instead of 5 supercoils. So that's topoisomerase 1, seeing that it only cleaves one of those strands. And then the net result is 1 less coil. Doesn't require energy because it's relieving the tension. Okay, now I need to be in a different mode here to show this. Yeah. Okay, so you've got, I think you've got that second part of that figure here. Let me just walk you through. So we're looking at topoisomerase 2 enzymes which introduce coils and generally introduce tension. But we are going to see in slides 15 or 16, there are 2 different orientations of coils. A positively directed coil and then a negatively directed coil. And so if you introduce a negative coil into a positively coiled chain, it actually relieves the tension. So there's an interesting example of introducing a coil requiring energy, but it actually relieves the tension in the strain because you've introduced a coil going in the opposite direction. So that's what is referred to here. It introduces tension if you introduce a coil that's going in the same direction. If you have a positively coiled chain and you introduce another positive coil, it is introducing tension. And I just say that because we're going to look at the opposite of that in slides 15 or 16. Okay, so the example here is the GRYB protein, standing for gyrase protein. And so here it is grabbing on. It has 2 different, it's a dimer, and within the dimer it has 2 different functional regions. And we're looking at the brown part of this enzyme that tethers, that grabs this enzyme. And you can see ATP is required to cleave, and we're actually cleaving both strands. So understand that this tube that we're looking at in this figure is a double helix of DNA, and when that whole strand is cleaved, you've cleaved 2 strands of DNA. And so then, once it's cleaved, it then winds the one part right through that cleaved part and then reanneals. So there's basically 3 steps. Cleaving of the double strand, feeding the loop through that double strand, and then annealing, gluing back together the 2 strands of DNA. And there is then the introduction of a coil. And that's the function of gyrase, the gyrase enzyme, which is a good example of the topoisomerase 2. And one of the things it has to do while it's doing this is keep those strands tethered. Otherwise it just flaps and you lose that coil. So I just point that out. So if the ends are tethered, it doesn't uncoil. Okay, and then here's a nice, do you have this figure in your, yeah, okay, so it's maybe just lower down. And you also have this text up here, right up here that I just, that I went over there? Do you have that on your outline? Okay, so that's meant for you to write down, okay? So do everybody have that written down? I'm about to cover it up. I didn't know how else to do this slide. Okay, so this is a really nice 3D. These are models that are getting close to reality as far as the actual physical shape and chemistry of these marvelous molecules. And so, so DNA is shown in green, and the 2 different parts of the molecule are shown in blue and red. And it is just in the process, and it has happened, of tearing those 2 strands, cleaving those 2 strands of DNA apart. And then it is in the process of looping the double strand right through this crevice right here is shown right through. So here is the crevice of that enzyme, and here it is about to be fished through. So, you know, incredibly concerted, coordinated action of this protein. You know, doing things that our hands would have to do. But it's at the molecular level. It's really, really cool. So 3D models give you an idea on the actuality as to how this is done, or certainly the physical conformation. So I just put that in there to, mostly for appreciation. You're certainly not going to have to, supercomputers write those models. Not quite, but it takes a lot of memory and a lot of data to construct these 3D models. So we're not doing that in this class, that's for sure. Okay, so here's kind of the heart of this chapter. And this is stuff that you know. So I'm not going to reteach you Okazaki fragments. I'm not going to reteach you the difference between the leading strand and the lagging strand. You have done that, okay? We are going to look at the biomachines and the names of them that carry the process out. But we do want to start on the same page. In bacteria, we also, I say we, they, you know, I relate a lot to them. They're my bread and butter. I love the bacteria. I used to carry pictures of them in my back in my wallet, and at one point my daughter said, "Would you carry a picture of me instead of the bacteria?" And so now, now I've kind of straightened my life out, thanks to my daughter. My sons didn't care. They said, "Oh, that's cool." Then my sons, when they were very, very little, one of them was crying in the car, and the 4-year-old says to the 2-year-old, he says, "Luke, what's the matter? Do you have bacteria on you?" You know, so that's the solution. If he's got problems, it has to do with what Dad works with. And he called them bacteria, so it's one of our cute family stories. But the boys didn't seem to mind me carrying pictures of bacteria in my wallet. Okay. So in bacteria, DNA duplication, DNA replication is semi-conservative, just like it is in our cells. So what do we mean by semi-conservative? Here is the original strand that needs to be copied in gray, and you have 2 replication forks, and both of them, you have then 1 new copy that is, of course, using the DNA sequence based in the original copy. So semi-conservative, the product conserves one of those strands, and now you have a new daughter strand. Just like you've learned in the past, bacteria carry out semi-conservative DNA synthesis, and just like in our cells, there's 2 replication forks going on. The synthesis is going on at the same time. So here is kind of the big picture of an entire chromosome that needs to be replicated. And there's this, what we call a replication bubble. So here is 1 replication fork, and here is a second replication fork. And just simply see how that bubble expands, and that replication bubble expanding represents the synthesis of more and more of that chromosome. Okay, so that's kind of the big picture view. You can see you have 1 original strand and 1 new daughter strand, and here at the end you've got duplicate chromosomes. You've got that 1 chromosome now synthesized into 2, 1 the original strand and 1 a new daughter strand. So again, no new information here for you. Those replication forks move in opposite directions. So this is kind of, you know, orienting you until before we get into the specifics. Okay, so this is my focus in this chapter is less on the figures and more on identifying these particular catalysts in these reactions. And it is kind of amazing to see 3, 6, 7, 8, 9, 10 separate biological catalysts. Biological, they're not particularly, you can't really call them motors. They don't have the rotor and the stator and axles like typical motors have, but they certainly are machines, biological machines, 10 of them, acting in concert with each other and working cooperatively together. It's just kind of mind-blowing, and we'll talk about the speed at which this is done, and then even the repair, the proofreading that happens. It's really it's a marvelous process. Okay, so these are the names of these enzymes that you should know and what they do, and we're talking about them in pretty basic terms. So here's your introduction: DNA, A, B, and C. DNA is the initiator protein. That doesn't seem very important relative to helicase. Okay. Helicase, what do you think the enzyme helicase does? I think I heard that. It digests the helix. So here you have a double-stranded DNA, and you simply have to open it up. So that is in a helix, and so helicase opens that helix up. And then you need, for that DNA, for that helicase to work, you need an enzyme to load it onto its substrate. So you need a helicase enzyme loader. DNA primase is the enzyme that initiates that little RNA primer on the lagging strand. And here is the workhorse of the process, DNA polymerase. And we're going to be looking mostly at the function of DNA polymerase 3. And so it polymerizes DNA. So look at the name, see what it does. Polymerase polymerizes DNA. Okay, now the DNA polymerase needs some help. So it needs a clamp loader, which carries the DNA polymerase 3, and the DNA polymerase 3 needs a sliding clamp in order to work. And so here is a loader that loads the DNA polymerase and the clamps and brings them to the DNA substrate. So you have a clamp loader involved in the DNA polymerase 3. And here is one that I'm probably in the end de-emphasizing, because I'm really de-emphasizing that lagging strand. We're not going to go into those details. But in order to get rid of that RNA primer, because by golly, we're making DNA, DNA polymerase 1 replaces that RNA primer now with DNA, and so we have a proper product of DNA. And then here's this topoisomerase 2 that we talked about, called DNA gyrase. So it actually, by introducing a negatively oriented coil into a positive coil of DNA, relieves tension. So it requires energy, but since the coil that it introduces is going in the opposite direction of the coils that are there, it actually relieves the tension. So DNA gyrase is important right at the front edge of the reactions. And so we're going to see where this happens, but one more list, maybe even two. Yeah, I just basically list 3 times these same catalysts, kind of describing them in 3 different ways. So this is where the heart of the information is. We're going to see in some figures that should help a bit. Okay, so DNA binds to DNA and initiates the process. Remember, it was called the initiator protein. Okay, DNA B is a helicase that unwinds helices. And it needs a loader, a protein that loads it onto its substrate. The primase synthesizes an RNA primer. So DNA polymerase 3, DNA synthesis and proofreading. And in order for DNA polymerase 3 to get onto the substrate, it needs a clamp loader which carries it, and this clamp loader also carries this sliding clamp. And the function of this sliding clamp is it tethers the DNA polymerase III onto its substrate, DNA. It clamps it on. So, part of my focus is just trying to transmit to you the mechanics of this process and the physics and the work that is done. Tethering, loading, clamping, these are mechanical terms. This is a mechanical, it's a biomechanical process. Okay, and then DNA gyrase. It relieves DNA supercoiling because as you are generating new DNA, it generates positive supercoils. And so the gyrase introduces negative coils to relieve this otherwise torsional stress. You could maybe think of DNA gyrase as Monk not letting supercoils be introduced because it just doesn't sit well with him. Okay? So relieving that torsional stress is the function of DNA gyrase. So then, after replication, any cleaved ends are ligated, are knit, sewed back together by DNA ligase. A lot of these proteins, you use their words, or the names tell you what they do. So if you know what the verb ligate, to ligate a wound, you know the function of a DNA ligase, is it's sewing it back together. Okay, we're going to probably finish here before we get into the actual figures. But let me walk through. This is my third slide. This kind of gut impression of what is going on. 10 different proteins just duplicating DNA so they can make a baby in another 3 minutes. It's this lust for life. All of them working at breakneck speed, covering for each other, helping each other. It's just, it gets a little anthropomorphic, but this is what's happening. So this is sort of like action words. So DNA binds, initiates, DNA B unwinds DNA, DNA C loads, this one primes, DNA polymerase, look at this, reads, inserts, proofreads. What is the proofreading capability? Well, it cuts out the incorrect base and inserts the correct one. Look at all those action verbs that one catalyst carries out. Clamp loaders carry and load. What about the sliding clamp? It clamps and tethers the DNA polymerase III to the DNA. DNA gyrase relieves torsional stress. You always want to have somebody on the site to relieve the stress that is generated in a very, very fast breakneck speed process. And then the DNA ligase. And this is happening at like 750 bases a second. Okay? All of these enzymes are working, just think about synthesizing, inserting one nucleotide. All 10 have to be working together to introduce one nucleotide. And there are 750 bases made per second in the concerted action of these 10, and there's actually more catalysts that are going on at the same time. So, really get an appreciation for this incredibly concerted, coordinated activity, and we will talk more about this on Wednesday, showing you some pictures. So start looking at those figures that kind of show these catalysts working together. So, see you Wednesday.